Hello, my name is Andreas, and I'm very excited to present our latest joint work on exploring automatic diagnosis of COVID-19 from crowdsourced respiratory sound data with myself and my collaborators, Chloe Brown, Jagmohan Chauhan, Jing, Apinan Hastanasombat, Dimitris, Spathis, Tong, Pietro, Cicuta, and Cecilia, Mascolo. Let's dive in. In this work, we employ crowdsourced audio data and show some preliminary evidence that cough and breathing sounds could contain diagnostic signals to discriminate COVID-19 users from healthy ones. We further compare COVID-19 positive user coughs against healthy coughs, as well as those from users with asthma. More precisely, our contributions are, firstly, an end-to-end description of COVID-19 sound collection framework through apps and the types of sounds harvested through crowdsourcing. Secondly, an illustration of the large-scale dataset that is being gathered. To date, this is the largest being collected and among the most inclusive in terms of types of sounds. It contains sounds from about 7,000 unique users, more than 200 of whom reported a recent positive test for COVID-19. Thirdly, we present our initial findings around the discriminatory power of coughs and breathe sounds for COVID-19. We construct three binary tasks. The first task aims to distinguish COVID-19 positive users from healthy users. The second task aims to distinguish COVID-19 positive users who have a cough from healthy users who have a cough. And the final task aims to distinguish COVID-19 positive users with a cough from users with asthma who report also having a cough. The results show that the performance remains above 80% area under the curve for all tasks. Finally, we show how audio data augmentation can be used to improve the recall performance of some of our tasks with less data. We see a performance improvement of 5% and 8% for task 2 and 3, respectively. Researchers have long recognized the utility of sound as a possible indicator of behavior and health. Purpose-built external microphone recorders have been used to detect sound from the heart or the lungs using stethoscopes, for example. These often require listening and interpretation by highly skilled clinicians, since audio signals contain rich and subtle information only able to be recognized by domain experts. However, recently and rapidly, these specialized recorders are being substituted by different technologies, such as a variety of imaging techniques, such as MRI or sonography, for which analysis and interpretation is easier. However, recent trends in automated audio interpretation and modeling has the potential to reverse this trend and offer sound as the cheap and easily distributable alternative. In this work, we focus on sounds generating by breathing, coughing, and reading that can be recorded from a commodity device at the patient's comfort, such as a mobile phone or a personal computer. This intuition stems from the recent observation that respiratory sounds collected by devices from patients tested positive for COVID-19 in hospital are distinctly different from sounds from healthy people. Our crowdsourced data gathering framework is comprised of a web app, an Android and an iOS app, along with the server infrastructure to back this up. The features of these apps are mostly similar. The user is asked to input their age and gender, as well as a brief medical history and whether they're in hospital. Users then input their symptoms, if any, and record respiratory sounds. They are asked to cough 3 times, breathe deeply through their mouth 3 to 5 times, and to read a short sentence appearing on the screen for 3 times. To ensure user privacy and data integrity, all transfers are encrypted with end-to-end encryption, and the files are stored in a secure, limited access server. Let us now explore the collection pipeline in a bit more detail. As discussed, the data collection pipeline is similar for all apps involved, and mainly it is comprised out of the following 3 discrete bits: the initial survey, the daily symptom survey, and the recording of actual sounds. The initial survey is completed only once for each user. The user is asked to input their age and gender, as well as provide a brief medical history. The daily survey asks the user their COVID-19 status, whether they are in the hospital, along with any symptoms present at the time of completion. Additionally, a location sample is optionally gathered with permission. This is followed by the recording of respiratory sounds. Specifically, the users are asked to cough 3 times, to breathe deeply through their mouth 3 to 5 times, and to read a short sentence appearing on the screen for 3 times. Finally, the data flows encrypted to our servers where it is stored securely. If a successful transmission happens, the data is then removed from the device. Helped by the organization of a large media campaign, we were able to crowdsource data from a large number of users. In particular, as of the 22nd of May 2020, our dataset is comprised out of more than 6,600 unique users that have donated at least one sample. Concretely, we have 4,352 unique users collected from the web app and 2,261 unique users collected from the Android app, comprising 4,352 and 5,634 donated samples respectively. Of these, 235 declared having tested positive for COVID-19, 64 in the web form, and 171 in the Android app. Of the Android users, 691 users contributed more than one sample, meaning they returned to the app after 2 days and reported their symptoms and sounds again. Let us now discuss the demographics distribution statistics. On the left figure, we see the country distribution as is inferred from the location sample. This shows that the most active countries in terms of overall contributions, named in increasing order, were Greece, United Kingdom, and Italy. However, we have to note that since location reporting is optional, many users opted not to record their location. The right figure shows the age breakdown, which can be seen that is skewed towards middle age, with the highest number of entries coming from participants aged between 30 and 39 years of age. Let us now discuss the self-reported symptoms. In this figure, we can see the most frequent symptom distribution for all Android users. We note that we do not know which users have or have had COVID-19 recently, but we do know that only a small fraction of these have tested positive, as discussed previously. In this group, the most common single symptom reported is a dry cough, followed closely by a sore throat. Interestingly, the most common combination of symptoms is a type of cough and a sore throat. Now, let us shift gears and narrow the scope of the symptom distribution evaluation down to the users self-declaring that they had a positive COVID-19 test. Interestingly, as we can see from the figure, the most common single symptoms are wet and dry cough, and the most common combination is lack of sense of smell and chest tightness. This is aligned with similar studies that have explored tracking of symptoms. The fact that cough is one of the most reported symptoms for COVID-19, but is also a general symptom of so many other diseases, provides further motivation for using sounds as a general predictor. Further, from here on, I will be passing the microphone over to my collaborator, Jing to conduct the rest of the presentation.
In this slide, an overview of our COVID-19 data analysis pipeline is presented here. And in the next few slides, more details for each component of this pipeline will be described. Note that while the data we collected are crowdsourced data, it can be quite messy and not many people have been tested. Hence, in our preliminary study, we analyzed cough and breathing data first and focused only on a curated set of the collected data. In our study, we are interested in distinguishing COVID-positive users from non-COVID users by using their cough and breathing data. For this aim, we manually checked all samples of users who said they were tested positive in the last 14 days or before, which gave us 141 positive samples for cough and breathing each. Then, as a control group, we selected users from countries where the virus was not prevalent at the time of data collection, such as Greece. And these users also have a clean medical history, never smoked, and no symptoms have been reported either. And this gives us another 298 negative samples. Furthermore, we also performed another two tasks specifically to analyze COVID-positive users who reported dry or wet cough as symptoms, and study if we can distinguish these users from non-COVID users who, however, also declared a cough as symptom, or further distinguish COVID-positive users with cough from non-COVID users who have asthma and a cough as symptoms. In this table, the number of samples we use are given. Besides, to process the data, we first resample all sync recordings to 22 kHz and remove the silence from the beginning and end of each recording. The second step is to extract features, and here we first extract 477 dimensions of handcrafted acoustic features, covering global features such as duration and period, and also features from frame level and segment level. For global features, after removing the beginning and the ending silence part, we extract the total duration from the whole signal as a global feature. Besides, the number of peak onset is estimated by identifying the peaks from an onset strength envelope. Also, we extract the main frequency of the envelope of the signal, which is the period attribute here. Then, for frame level features, we extract features such as root mean square energy, spectral centroid, roll-off frequency, and zero crossing rate of the signal in each frame of 100 milliseconds, and then use a sliding window to compute the feature over a sequence of frames. Also, we extract the MFCCs as our substrate features. In particular, we use the first 13 MFCC components and its first and second order of the temporal differential information. Then, aiming at creating segment-level features to present the whole instance, we applied 11 statistical functions to describe the distribution of each frame-level feature. These statistical functions including mean, standard deviation, median, minimum, maximum, and etc. Moreover, we also consider to deploy transfer learning-based features or so-called data-driven representations. Particularly, we use the pre-trained VGG-ish model by research group of Google, which is pre-trained by a large audioset database for audio analysis. The model is a CNN-based architecture which first divides data samples into short subsamples, and for each subsample, it returns a 128-dimension feature vector. Again, we take the mean and standard deviation across the entire sample as our final features, and therefore it has 256 dimensions of each. Besides exploring these two feature sets separately, we also use the early fusion to combine varied features, and the final feature vector can be up to 733 dimensions. Then, for feature reduction, we applied PCA. Then we trained SVM models for the three aforementioned binary classification tasks and investigate the performance of varied feature types and modalities. In particular, we ran the multiple runs, and for each run, the data were split into disjoint training and test split with a ratio of 4:1. Then, for optimization, we carried out 5-fold cross-validation on the training partition to optimize the hyperparameters to find the best error and curve performance. And for performance measurement, we report our results in terms of error and curve, precision, and recall respectively. Next, in this table, we report the classification results in terms of AUC, precision, recall for 3 binary classification tasks separately. Both the mean and standard deviation over 10 runs are listed here. It can be seen overall we have achieved around 80% of AUC for each task and around 70% for precision and recall here. Then, when further looking into the best modality selected for each task, it has been shown that the best performance for each task is obtained from different modalities. For instance, for task 1, the best performance is achieved when using both cough and breathing to distinguish COVID-positive from non-COVID healthy user with a clear medical history and no symptom reported. However, for task 2, it has shown that cough signals are able to distinguish COVID-19 positive cough from the non-COVID cough quite well. When it comes to COVID cough with asthma cough, it's interesting to see here that the breathing sounds serve as more powerful signals to discriminate users here. Moreover, when comparing the different feature set types, it has been shown the feature type 2, which is the VGG-ish feature, in most cases performs better than the handcrafted feature, which is feature 1 here. This shows the promising of using transfer learning in our analysis. And last, from our results, one can see the early fusion of handcrafted feature and VGG feature, which is the feature type 3, provides the best results in most cases. This indicates that the two different feature types provide complementary information from the signal, or saying that they present the original signals from different aspects. Further, to counter the small amount of control data available for tasks 2 and 3, we augmented the negative samples for these two tasks using 3 standard audio augmentation methods, which is by amplifying the original signals, or by adding white noise, or by changing the pitch and speed. We apply this method twice, and this results in 6 times in total the number of negative samples for task 2 and 3 respectively. Note that we only use the augmented samples for training, not for test. From the result table, we can observe that the performance in terms of AUC and recall got almost a 10% increase when compared with the previous result. Also, we achieved a slightly smaller standard deviation. To conclude, in this paper, we have presented an ongoing effort to crowdsource respiratory sounds and study how such data can help COVID-19 diagnosis. In particular, we use machine learning approach to distinguish COVID-positive users from 3 different non-COVID user groups by exploring their cough and breathing sound. Our experiment results have also demonstrated the advantage of using transfer learning-based deep features and data augmentation for COVID-19 sound analysis. In future, we plan to continue collecting more data to improve the accuracy of our model. Also, our mobile app reminds the users to provide samples every couple of days, As a consequence, we may have a number of users from whom we could study the progressing of respiratory sound in the context of this disease. This is something we have not yet investigated in the current work. Then, we also plan to look into the speech data and integrate different modalities to improve the performance. And further, we plan to introduce other advanced features and deep learning approach specifically for COVID-19 sound analysis. Last but not the least, you are welcome to scan this QR code here, which will link to our homepage, where you can download the app or find our recent research findings. Thank you for listening, and please donate your data and save lives.